train in your lab. I think his lab has really done an amazing job of engaging that next generation of scientists and giving them opportunities to get excited about neuroscience. So please join me in welcoming today's speaker, Geoff Donlea.
Thank you, everybody, and thanks, Lindsay, for the kind invitation to speak today and the really nice introduction. So as Lindsay mentioned, ever since I started grad school, I've been interested in using small animals to try to understand sleep. I think it's a question that I've been interested in because, I'm sure everybody in this room knows what it feels like and the impacts of not getting enough sleep on our own daily lives. And that's probably an experience that we share with just about every other type of animal in the world. Right? So sleep is something that's tightly evolutionarily conserved across all animal species. And insufficient sleep is a real public health concern that can have profound impacts in different workplace environments and then out on the road in the world around us. Aside from just these kind of narrow contexts, we think that there are probably tens of millions of Americans who chronically don't get the sleep that they probably need and may be suffering from negative health consequences as a result. Right? And so because sleep is something that's so tightly evolutionarily conserved, and because just losing a little bit of it can have a large impact on how we function, the field has kind of coalesced around this idea put forward by Alan Rechtschaffen in the 1980s that sleep must serve some absolutely vital function or set of functions, or else it's been a huge waste of all of our time. And so my lab uses simple fruit flies to try to understand what these basic functions are and to try to pick apart mechanisms that underlie what sleep does to maintain health in the body and the brain. To measure sleep in flies, we take advantage of a really simple behavioral assay that is high throughput and very scalable. We take individual fruit flies and put them in these little glass tubes that are about 3 or 4 inches long. We put fly food at one end of the tube and then seal it with some melted wax, and then a foam plug in the other end of the tube so the fly doesn't escape. And then we put these individual tubes in these activity monitors that have an infrared LED on one side and then a photosensor on the other side of the tube. So every time the fly wakes up, it alternates between usually eating a little bit and then patrolling the length of the tube looking for an escape route. And we can measure that activity by counting beam breaks automatically every minute over the course of a several-day-long experiment. Okay. And so we look for periods of inactivity that last for at least 5 minutes. And those quiescent periods match all the behavioral properties that we would look for in sleep in any other species. These flies are quiescent for a period of time. They have increased arousal thresholds after several minutes of inactivity, but the state is rapidly reversible, so we can wake up a sleeping fly on demand. More recently, we've also begun to use activity monitors that instead of having one infrared beam that bisects the tube, we have new monitors that use 15 infrared beams across the length of the tube to get a much higher spatial resolution to make sure that we're not missing low levels of activity in some of the genotypes that we're interested in. Okay, and this type of monitor will come into play in the projects that I described, especially in the second half of my talk. All right, so if we put our flies in our activity monitors, we usually put them on a 12-hour light, 12-hour dark schedule. We can see that our flies are mostly, and then we can measure sleep in minutes per hour on the y-axis here. You can see that our flies are mostly awake first thing in the morning when the lights turn on, and mostly awake in the evening right before the lights turn off. They have 2 large activity peaks during these time periods. In the middle of the day, they have a nice little siesta period right after lunch, I guess. And then once the lights go out, the flies fall asleep and then stay asleep for most of the night. These activity patterns are stable within an individual and then reliably consistent across populations of flies from batch to batch. Okay. And so for the rest of the talk, rather than focus on one particular recent paper in my lab, I wanted to kind of take a broader approach and give you a big picture run through of a couple of projects that have been running in parallel in my lab, trying to examine sleep regulation and function on different timescales. The first of these has examined the impact of manipulating sleep history on synaptic organization in the brain using Drosophila melanogaster as a nice genetic model that we can exploit all the genetic tools that are available. And in the second project, we've taken a little bit of a broader approach to examine evolutionary trends in sleep patterns across related fly species. Okay, so without delay, I'll jump right into this first project. So for the past several decades, there's been a big interest in the sleep research community on trying to understand the impact of changes in sleep on brain connectivity and plasticity. And a lot of these studies were motivated by findings that sleep deprivation can rapidly impact cognition, including learning and memory. Right? And so a few key studies using the fly as a model have started to find broad trends in synaptic scaling across the fly brain when you deprive flies of sleep. In this study from Giorgio Gillestro, when he was in Chiara Torelli and Giulio Tononi's lab, as a postdoc in Wisconsin, Giorgio deprived flies of different amounts of sleep and then took whole brains out, homogenized them, and then used Western blots to quantify overall changes in core synaptic protein markers. I want to hone in on this particular protein called BRP, or Brook pilote, which is a core structural anchor protein for the presynaptic active zone, and is kind of the best surrogate marker that we have in the fly for synaptic strength or abundance. And as you can see, as you sleep deprive flies for 6, 12, or 24 hours, you see this scalar increase in BRP across whole brain homogenates in the fly head. Follow-up studies have tried to use immunostaining to quantify protein levels across whole fly brains. This is a study from Stefan Siegrist's lab, published more recently. And they found that if they stain for BRP, or one of its core interacting proteins on the presynaptic active zone, called DSID1, you can see that these chronically short-sleeping insomniac flies have stained for more BRP and DSID than wild-type controls. Okay, so it looks like there's this broad synaptic scaling that happens across the brain in flies as they're awake for more time than they're used to. And so these data points and several others have kind of led to the development of this hypothesis that typically we can navigate the world and encode new information by forming new synapses and expanding existing ones in the brain. This is great because it allows us to adapt our behavior to best fit the changing world around us. But if allowed to go unchecked, eventually this kind of chronic synaptic upscaling is going to hit some ceiling in terms of either space or energetic constraints or synaptic saturation. And so in order to kind of keep some kind of synaptic homeostasis, there must be some kind of downscaling that kicks in during sleep that allows the brain to retain relative changes in synaptic weights that encode important information that we learned earlier, but allows some capacity for new learning the next morning. So a few studies have tried to examine this type of hypothesis, and I think the fly is going to be a really good model to dig in and identify mechanisms that drive synaptic scaling on different time courses in the brain. Okay, and so we've taken a couple of different strategies to try to identify trends and mechanisms that kick in to scale synapses during sleep. The first one is to identify acute experiences that we can deliver to flies that rapidly change their need for sleep, and then dig into those acute time periods and understand the functional role of sleep in synaptic turnover in those conditions. Okay, and so it's just a few examples. We know that things like changing the social environment that a fly is housed in, or giving flies a physiological stressor like a brief heat shock, or even giving flies these somnogenic drugs like this GABA-A agonist called carboxyl, can rapidly drive an increased need for sleep. And we're kind of on patrol in my lab to look for other contexts that also drive similar changes in sleep. And one that we've become really interested in over the past few years is this really simple neural injury model where you take the flies and then anesthetize them on a CO2 pad, and then remove the exterior segments of their antenna with fine forceps. Now, this type of antennal injury has been studied for almost 20 years at this point, including a lot of really nice work from Mark Freeman's lab, initially at UMass and more recently since his move to Oregon. And they found that if they genetically label subsets of olfactory receptor neurons that are severed by this type of injury, you can label their plasma membranes with CD8 bound GFP, and you can see this highly programmed Wallerian degeneration process that kicks in over a few days after injury that actively eats up the plasma membrane from these severed cells and completely clears it from the brain. We became interested in trying to understand if sleep plays a role in this type of clearance mechanism. And so we took some wild type flies and had a group that was going to be injured in blue and some control siblings in gray. Both groups sleep exactly the same on a baseline day before injury. You can see that we delivered injury on the second morning, and the injured flies sleep much more than their handled control siblings. And then by the next day of recovery, we can see that both groups are back to the same levels of sleep again, indicating that we haven't permanently broken sleep in these injured flies, but there's some acute response during this first day after injury that may be worth digging into in more detail. We initially wanted to make sure that these flies were actually sleeping and not in some other kind of quiescent state. And so we took our activity monitors and strapped them on the top of a vortexer and delivered a mild shake. And we found that our control flies rapidly woke up with a 0.5 G stimulus, while our injured flies were able to sleep right through that mild shake. But if we cranked up the dial a little bit, then all of the flies woke up and started to run around. So we think injury is putting these flies in a sleep state that's deeper than control flies would typically experience. But these flies can be rapidly woken up, indicating that it is sleep and not some other state. To back up that conclusion, we also tested post-injury responses in a couple of chronically short-sleeping mutant flies, both for the carrying alleles for insomniac gene and sleepless. We can see that these short-sleeping mutants really don't show any increase in sleep in the first few hours after injury, while genetic controls show large increases in sleep right after injury. So we think that the same genetic programs that drive daily baseline sleep regulation also kick in to promote sleep after injury as well. We wanted to try to understand the role of injury responses in sleep regulation. So we wanted to test whether other types of injuries might also change sleep. So as I showed you, we've already severed antenna. We also wanted to, we tried to cut wings off flies. We severed maxillary palps, which are a secondary olfactory organ. And we also removed halteres, which are a proprioceptive organ that are basically an external vestibular system for the fruit fly. And we saw different types of responses. So interestingly, we saw an intermediate response if we severed one antenna instead of two, indicating that there might be some dose response in terms of the degree of the injury and predicting how much sleep they were going to get afterwards. When we severed the wings, we saw a significant increase in sleep during the day, but we saw no response when we severed either maxillary palps or halteres. So it looks like there's a wide range of responses in sleep after different types of injury. We're still trying to uncover the rules that govern the sleep responses, but it's tempting to think that the number of injured sensory neurons might predict how much sleep these flies are actually going to need in order to recover. Okay. So at that point, we concluded that sleep is increased after injury. We were really interested in the functional importance of what that sleep was doing to aid injury response and recovery afterwards. But we were put in a kind of a condition where we were really interested in that first few hours after injury, and there hadn't been much of a characterization of what kind of degradation happens during that first few hours. Many of the previous studies had focused on at least a day after the injury. And so we were interested in trying to label presynaptic active zones in these ORNs and their plasma membrane after injury. And so we had really good genetic tools to label these drivers or to label these cell types. And then great neighbors across the street in the Zapersky lab who had recently created this synaptic labeling tool called STAR that allowed us to tag endogenously driven BRP expression in active zones of these cells of interest. And so we injured flies and labeled plasma membrane with CD8-GFP, and then presynaptic active zones by histologically tagging this core synaptic protein that I mentioned before called BRP. And what I hope you can appreciate is that BRP is cleared much more rapidly than the plasma membrane itself. And the rate of BRP clearance almost really closely matches the time course of the increase in sleep that we see during the day. So you see a lot of sleep happen for about 9 hours or so, and by that point, a lot of the BRP is already removed. So our next question was whether sleep plays an important role in facilitating this BRP removal. And so we repeated this experiment, but allowed half of our flies to sleep and sleep deprived the other half. And we can see that the flies that were allowed to sleep clear out a large amount of BRP after injury, while the flies that were kept awake retain a much larger amount of the BRP in these damaged axons. So we're really interested in trying to now understand the acute mechanisms that kick in during this first 6 or 9 hours after injury to both drive an increase in sleep and to promote BRP removal. So there's a few prime candidates that we're trying to track down now. The first one that we wanted to test were these microglia-like cells called ensheathing glia that typically kind of surround neuropil compartments, and then had previously been found to be the primary phagocytes that invade neuropil after injury and start to eat up the fragmented axons during days after injury. We found that these cells infiltrate neuropil at 24 hours after injury, but are not there at all by 6 hours after injury, which is the time at which we see most of the synapse clearance. So we think that there are other mechanisms waiting to be uncovered that kick in during this acute time period right after injury before ensheathing glia are activated in order to both promote sleep and clear out synapses themselves. So we're actively trying to figure out those mechanisms. The second strategy that we've tried to use to understand the impact of sleep history on synaptic organization is by simply sleep depriving flies and then measuring changes in synaptic reporters in different circuits of interest. Right. And so just to remind you, these initial studies early on that found broad changes in synaptic proteins measured either whole brain homogeneous or a whole brain fluorescent signal from immunostaining. And we're really unable to tell us whether these changes in BRP were spread evenly across different cell types or even across different brain regions, right? And so we thought that in order to identify the mechanisms that were driving this kind of synaptic scaling, we first had to figure out which cell types might be most sensitive. And so the first grad student to join my lab, Jackie Weiss, turned to measuring these trends in the mushroom body. This is a core associative memory center in the fly brain. And efforts over the past several decades, culminating with large studies driven by the Jerry Rubin's lab at the Donlea Research Campus, have generated great genetic tools with single cell resolution to label and manipulate these cells. We think this is a great place to look because sleep is important for mushroom body dependent learning and memory. This is an example trace from an olfactory assay that I used when I was a postdoc in Gero Miesenbach's lab. We put flies in a small chamber in which they had 2 odors puffed in on either side. They could then choose to spend their time in either odor. You can see that these rested flies during this pre-training test preferred to spend their time in this side of the chamber represented by the blue odor. We then paired that blue odor with an electric shock, and then these flies were pretty good at being able to learn to avoid that odor and shift their preference to the other side of the chamber. If we took siblings that had been sleep deprived the night before and put them through the same task, we can see that these flies were able to perceive the odors in the same way initially. They have the same initial preference for this odor on the left side and represented by blue. We then paired that odor with an electric shock, just as we did for their rested siblings. But these sleep deprived flies didn't get the lesson and were unable to shift their preference after the training. So we think that there's some deficit in mushroom body dependent odor processing and learning and memory. So in order to assess whether synaptic organization changes in the mushroom body, we first turn to a protein trap reporter for BRP that labels BRP in every cell in the brain. Jackie dissected brains from either rested or sleep-deprived flies. And as we might have predicted, you can see a large gain in BRP across all of these neuropil lobes of the mushroom body. The magnitude kind of surprised me. You see like a 25 or 30% increase in this core presynaptic active zone protein, basically everywhere in this memory encoding circuit. We also had a nice thermogenetic tool that we could activate some sleep-promoting neurons using a warm-sensitive cation channel and force flies to take a nap for a few hours. And we saw after this nap, the flies had the opposite effect. That these experimental flies where we induced sleep for about 6 hours lost about a third of their BRP in the mushroom bodies, while genetic controls that had either not been heated or that did not express the sleep-inducing cation channel had no change in BRP. So we think that there's a bidirectional effect of sleep history on synaptic organization, where the longer you're awake, there is more BRP accumulating in presynapses, and with more sleep, either recovering from sleep deprivation or artificially induced, these cells clear out BRP. The next step was to try to identify whether these changes in BRP could be attributed to particular classes of neurons in the mushroom body. And so we used genetic drivers that had already been characterized, first to label presynaptic BRP in excitatory mushroom body intrinsic Kenyan cells, or KCs. The output synapses from these cells are the sites of learning and memory for olfactory assays. And we saw that after sleep deprivation, these excitatory cholinergic Kenyan cells show a large increase in BRP across all lobes of the mushroom body. And then if we allowed flies to recover for a couple of days, eventually BRP levels came back to control levels again by about 48 hours with extra recovery sleep. In contrast, we saw either small local changes or no changes at all if we labeled presynapses in either punishment encoding or reward encoding dopaminergic neurons, or if we labeled large recurrent interneurons that provide lateral inhibition across the whole mushroom body. Right, so we think that the excitatory cholinergic Kenyan cells are the class of neurons that's driving almost all of the BRP accumulation across the mushroom body as flies are sleep deprived. And so we're really interested going forward in trying to understand why this particular class of neurons that's especially important for learning and memory might be sensitive to upscaling their presynapses with sleep loss. We're interested in trying to understand whether there are particular subsets of synapses within these cell types that might be especially sensitive, and whether recent activity or recent plasticity might either be protective or line these cells up for more plasticity during sleep loss. And we're interested in trying to understand which mechanisms reduce presynapses in Kenyan cells during sleep. Over the longer term, we're especially interested in trying to pick apart the mechanisms that we find from our injury project and trying to understand whether those synaptic pruning mechanisms that might be active during sleep could be applied more generally to help other circuits recover from sleep loss. Okay. So we've had a pretty deep dive into the mushroom body, but we were really interested in trying to understand whether similar trends might apply throughout the rest of the brain as well, right? And so Jackie Weiss, who's the grad student who had taken on the mushroom body project, by that point had to manually dissect, I don't know how many flies, and then analyze each brain on their own. And so we're trying to look for a higher throughput approach to assay trends in other neuropil regions as well, instead of having to go region by region individually. And so we took a step back and started to take our presynaptic labeling tool that was shared from the Zapersky lab to label BRP and cell types of interest and express it across neurotransmitter systems throughout the whole brain. And so we labeled presynapses in excitatory cholinergic cells throughout the brain. In the fly, acetylcholine is a primary excitatory fast neurotransmitter. We also labeled presynapses in glutamatergic neurons throughout the brain, in dopaminergic neurons and GABAergic neurons as well. Okay. And then we dissected whole brains from each of these groups after either a night of sleep or a night of sleep deprivation, collected whole brain confocal stacks to image BRP distribution in every neuropil region in the central brain. And then instead of analyzing them one by one, we aligned each to this common brain template that had been developed by at Donlea by the Rubin lab, which also came with an automated mask that allowed us to quantify BRP changes across about 36 different neuropil regions in the central brain. Okay, and so this sped up our analysis a lot and allowed us to collect whole brain stacks through a couple of dozen brains in each one of these groups, and then rapidly align them and do local quantification. And so these are averages of control in sleep-deprived brains. The heat map is intensity of BRP labeling throughout the whole central brain of the fly. And so if we do a Z projection, you can pretty quickly see that there are warmer colors in the sleep-deprived brain compared to the control when we label all cholinergic presynapses. And on the right, when we quantify mean intensity across the whole brain, there's about a 30% increase when we label cholinergic presynapses. Okay. In contrast, when we look at either dopaminergic, glutamatergic, or GABAergic synapses, we see either no increase or only a mild increase of a few percentage points. So this tells us that perhaps the trends that we saw in the mushroom body might not be particular to the mushroom body. They might be shared throughout the whole brain. We then dug in another step and automatically segmented each one of these whole brain images by about these 35 different neuropil regions. And much to my surprise, we saw that there was almost a uniform increase in cholinergic presynapses across the whole brain. I was expecting perhaps that places like the mushroom body where we'd already looked would be perhaps hotspots for plasticity, especially since they're known to have a role in associative learning and memory, and that other areas, including primary sensory areas, might be a little bit more stable, but that's not what we saw. When we did our local segmentation across the other neurotransmitter systems, we saw no local changes across dopaminergic or glutamatergic neurons, and then only a small increase in one local neuropil region in GABAergic cells, indicating that indeed the trends that we saw in the mushroom body may be applicable across the whole brain. And so because we have really precise tools to label and manipulate individual cell types in the mushroom body, hopefully we can find mechanisms that drive synaptic scaling in that circuit and then apply them more broadly in other circuits as well. Previously, earlier studies, as I showed with the Western blots from the Chiarelli lab, found a kind of a stepwise increase with longer sleep deprivation. So we also wanted to deprive flies for a little bit longer and see if there were knock-on effects on other neurotransmitter systems as well. And so we did 24-hour sleep deprivations when we labeled dopaminergic, glutamatergic, or GABAergic synapses. And indeed, we saw that after 24 hours of sleep deprivation, we saw a gain in glutamatergic synapses basically uniformly across the brain, although we saw no change at 12 hours. And then dopaminergic and GABAergic synapses remained relatively stable even after 24 hours of sleep deprivation. Okay. And so through these plasticity studies so far, we can conclude that sleep is temporarily increased after antennal injury. This is an acute experience that we can use to drive rapid and local synaptic clearance. And if we take sleep away from flies, the removal of presynapses from severed OR ends slows or even stops. And we're digging in now to try to use this assay to identify molecular mechanisms and cellular mechanisms that kick in during the first few hours after injury in order to promote sleep and selectively remove synapses from damaged axons. And then more broadly, in other contexts of sleep deprivation, we can see that there are broad gains in presynaptic BRP, which is a core presynaptic active zone protein in specific neuron classes. That we see overnight sleep deprivation driving first a gain in more BRP and excitatory cholinergic cells. And then if we extend the sleep deprivation even further, we start to see increases in other neurotransmitter systems in glutamatergic cells. And we're still trying to understand whether these waves of plasticity might reflect either compounding effects as more and more cell types might start to malfunction, or whether they might reflect layers of compensatory adaptation as glutamatergic cells might try to compensate for elevated cholinergic release or something like that. And then finally, these BRP changes are broadly shared across all neuropil regions in the central flybrain. And this gives us a kind of a hope that if we hone in on particular circuits where we have the best genetic tools, that we can identify the mechanisms that locally drive this type of homeostatic plasticity during sleep or sleep deprivation, and then plug those mechanisms into other circuits as well to try to locally rescue the consequences of sleep deprivation. Okay. So hopefully that gives you an overview of the types of approaches that we're using to understand the consequences of sleep loss in terms of brain plasticity. Right. And now I'd like to turn and change gears a little bit. Oh. Okay. To go from kind of acute responses of sleep to longer term adaptations that may have occurred over evolutionary time in millions of years. Right. And so I started off by showing you this slide that and making the point that sleep is a state that is broadly conserved across evolution, right? We can see it in pretty much any animal species that we want to study. But we all know that even though sleep is conserved, it occurs very differently and on very different timescales at different species. Anybody who's been to the zoo knows that there are particular animals that you know are probably going to be walking around and eating for most of the day. And then other ones like lions and tigers that you're most likely going to have to look for, they're going to be napping in the shade back in the corner somewhere. And so different groups have tried to observe sleep patterns across all kinds of different vertebrate species. And a few really careful regression analyses have tried to identify different trends in either physiology or behavior or body characteristics that can predict changes in sleep time. But going from these linear regression trends to actual mechanistic studies is very difficult when you're measuring sleep in things like elephants or bats or other species that you can't really do scalable controlled studies on in the lab. And so I think this is an approach where invertebrates might give us a really promising lead. And so we've started to compare sleep in different fly species. Behavioral geneticists have focused on Drosophila melanogaster as a key model system for almost a century now. But if we look a little bit more broadly, we know that there are at least 4,000 other Drosophila species out there in the world. And so as a proof of principle, we've kind of focused on a couple of species that we can find locally in the area around us. Right? We know our good friends Drosophila melanogaster basically live wherever there are humans at this point, that they live off of our trash or our fruit orchards, and basically live off of the fruit that we eat. And if we drive outside of LA just a couple of hours to Joshua Tree, there's a completely different Drosophila species called Drosophila moavensis that lives in the cactus plants that are native to the Mojave Desert. So we got a stock of these Drosophila moavensis into the lab and threw them into our sleep activity tubes. And even without quantification, you can notice that there's obvious differences in the behavior of these flies, right? The Drosophila melanogaster on the left here, you can see most of them walking around after we just loaded them into the tubes. And just a few minutes after we put these moavensis into the tubes, you can see many of them have already kind of settled down and are standing still and look like they're taking naps already. If we look at behavioral trends over the course of the day, we can see we put our flies in a 12-hour light, 12-hour dark schedule. The reds represent 2 wild-type stocks of Drosophila melanogaster that we typically use as genetic controls in the lab. And then the blues show sleep traces from 4 different subspecies of Drosophila moavensis from geographically isolated desert regions. And what pops out right away is that these desert-adapted flies sleep a lot more, both during the day and during the nighttime, compared to Drosophila melanogaster. Initially, we were a little bit concerned that we needed to kind of do a finer analysis to make sure that this was actually a state that would match the behavioral properties of sleep in melanogaster. And so we quantified the length of sleep bouts during the day and the night. You can see that many of the sleep bouts during the day and the nighttime, and these desert adapted flies are much longer than they are in Drosophila melanogaster stocks. In studies of Drosophila melanogaster sleep over the past 25 years, we've typically used a minimum threshold of 5 minutes of quiescence as a marker that the flies had fallen asleep. We thought that maybe this 5-minute threshold wasn't strict enough for Drosophila moavensis, but we found that if we extended that minimum threshold out even to an hour, we can see exactly the same difference in sleep between melanogaster and moavensis. Okay, so for the rest of the studies, we continued to use this 5-minute threshold, just as we typically do for melanogaster. I was also concerned when we got these flies in that maybe this high sleep response was just an initial acute response to trapping the flies in a tube for the first time. So we continued to measure sleep over several days. Each row represents an individual Drosophila moavensis, and then these heat maps represent activity patterns over the course of a week. You can see that these daily patterns typically repeat for each individual fly, suggesting that this high sleep pattern is really stable over time and the way that these flies actually behave. We were also worried that maybe these flies weren't actually sleeping, but they were just kind of hiding out at the food end of the tube and maybe binge eating all the time. So we obtained some liquid food activity monitors. Each of these raster plots represents a meal from an individual fly over the course of a 24-hour day. And you can see that these melanogaster typically eat a lot during the daytime, and then they fall asleep and stay asleep at nighttime. But the feeding patterns of these Drosophila moavensis are much more sporadic. They eat dramatically less than melanogaster do, but when they get a snack, they typically eat for a little bit longer. Okay. So we think that, if anything, their food intake is dramatically reduced compared to Drosophila melanogaster. We also ran a few other control experiments to confirm our interspecies sleep differences. We obtained independent stocks of Drosophila moavensis from an entirely different source and found that they also show a large increase in sleep compared to our melanogaster controls. We were worried that maybe the fact that our melanogaster wild-type stocks had been housed in the lab for decades at this point might have impacted the way that they sleep. So we went out and caught a new wild-derived strain of stock of melanogaster from the neighborhood here and found that they sleep just like the flies that we've been using in the lab for a long time. And we were also worried that maybe our local food diet that we cook here at UCLA might not be healthy enough for these Drosophila moavensis. So we also reared them on an alternative diet that contains cactus extract from the plant that is their usual host. And we found that the sleep trends still hold up. Finally, another study had shown from the Marco Gallio's lab had also been studying these Drosophila moavensis in parallel and found that their temperature preference is different than melanogaster. They prefer to be in a slightly warmer environment. And so we ran our sleep assays at different temperature steps and found that it really didn't impact the way that these moavensis slept. We were interested in trying to understand whether this is a unique adaptation of this one species or might be common across other desert adapted species. And so we looked in the phylogenetic tree and found that there are a few other species of Drosophila that are also adapted to different desert environments. And we found that their sleep patterns are also elevated very similarly to Drosophila moavensis. So this may be a common adaptation of life in the desert. So these flies, they sleep a lot. The next question was to try to understand whether they would homeostatically defend this baseline sleep or not. You can kind of imagine that maybe these flies are just a little bit lazy, and if they don't have a reason to move around, then they don't. And in that case, maybe if you sleep deprive them, they wouldn't need to make up the sleep that they lost. Another possible alternative is that these flies have a high homeostatic set point, that they actually need all the sleep that they're getting. There's some functional benefit to it. It's adaptive, and they will actually recover it if you sleep deprive them. And so we took our flies and put them in our activity monitors and then sleep deprive them by stimulating them on a vortex each minute over the course of the night. And we can see in our Drosophila moavensis stocks that they show an increased sleep on the first several hours of recovery compared to what they would during baseline, indicating that they do have a significant homeostatic response to sleep deprivation. So in some cases, you can see that the response in sleep time isn't as big as what we usually see in Drosophila melanogaster, likely because they're sleeping so much to begin with that there's not a whole lot of extra room for them to show more sleep. So we probe the sleep depth in these flies while they're rebounding by housing them in constant darkness and then shining a light at them every hour or so. We can see that the flies that had recently been sleep deprived were less likely to wake up than either rested controls or flies that had been allowed 24 hours of recovery after sleep deprivation. And then when these flies woke up in response to an LED stimulus, they fell asleep much more rapidly when they were sleep deprived compared to the other two groups, indicating that these flies are rebounding and responding to sleep loss both by sleeping more and by sleeping more deeply than they would otherwise. Okay. So, at this point, we were really interested in trying to dive into neural mechanisms that were contributing to these changes in sleep patterns, right? But we were really used to having all of the genetic tools that we have in Drosophila melanogaster, and we're basically starting from scratch with this new species. So we were trying to understand where to go from there. And we turned to some great neighbors down the hall who run a mass spec lab. So we teamed up with Ghazi Alezi and Julian Whitledge and Semmel to do mass spec for different neurochemical modulators in the brains of these flies. And so we ground up heads from Drosophila melanogaster and Drosophila moavensis, and then used mass spec to measure relative abundance of serotonin, octopamine, dopamine, and histamine in the heads of these flies. You see that there's not really a big difference between the species in dopamine or histamine levels, but there was a really dramatic increase in serotonin and a loss in octopamine, which is the invertebrate analog to norepinephrine in the fly. And so both of these are interesting to us in terms of sleep. We've tried to dig in a little bit more to what these might mean. If we stain for serotonergic neurons by immunostaining for the serotonin reuptake transporter in both melanogaster and moavensis, you can see that the same cell types that are present in melanogaster are also there in moavensis. So there's not an obvious change in the organization of these neuromodulatory systems. But we're really interested in trying to understand whether the increase in serotonin in these species might contribute to some of the sleep phenotypes based on a project that is already ongoing in collaboration with David Krantz's lab in Semmel, where we found that either genetically lesioning the serotonin reuptake transporter or by pharmacologically inhibiting the serotonin reuptake can promote sleep in melanogaster. So we're trying to understand both the circuit basis of these phenotypes in melanogaster and seeing if we can transfer those same mechanisms to moavensis to understand adaptations in that species. We're also interested in trying to understand whether changes in this levels of octopamine also contribute to the high sleep phenotypes in Drosophila moavensis. Cesar Nave in the lab did a really careful job of immunostaining for this biosynthetic enzyme called TDC2 that labels octopaminergic neurons. You can see that many of the cell types are also present, that are in melanogaster are also present in moavensis, except for this one cell cluster in the anterior medial portion of the brain called ASM, which is present in melanogaster, and either very weakly or doesn't label at all for TDC2 and Drosophila moavensis. And importantly, studies from Amita Segal's lab have found that activation of these ASM neurons strongly drives arousal in Drosophila melanogaster. So Cesar took on a brave project to try to understand whether the downstream targets of octopaminergic systems might also promote arousal in Drosophila moavensis. And he took moavensis and did nano injections of octopamine into the central brains of these flies. And you can see hints that they significantly lose sleep over the next day or so and show increased locomotor activity in response to octopamine injections. So this is a hint that maybe This desert adapted species has lost arousing octopamine signals, but that the downstream targets might still latently remain inactive. Okay. The next step was to try to understand whether there's any functional relevance to this long sleeping phenotype in these desert adapted flies. Right? These flies live in an environment where you have large temperature fluctuations, and then food may not be available all the time. So we wanted to try to test whether their sleep phenotypes might contribute to resilience to starvation and food deprivation. And so we took flies and either food or food and water deprived them. And we can see that in red, our Drosophila melanogaster stocks start to die out after just a couple of days of food deprivation, while our desert adapted flies last much longer. Previous studies had shown that if you food deprive Drosophila melanogaster, they basically wake up and lose a lot of sleep to forage until they starve, while our desert adapted Drosophila melanovensis stocks basically hunker down and show very similar levels of sleep, whether there's food available or not. In order to test whether this retention of high sleep enables these flies to last longer under conditions of starvation, we took them and either housed them in their standard LD conditions or constantly shined blue light on them to disrupt and suppress sleep. We saw that our blue light exposed group died more rapidly from starvation compared to our LD controls. And when we did an individual plot of daily sleep in hours per day versus survival time, you can see a strong positive correlation between the two, indicating that higher levels of sleep may be protective for these flies and enabled them to withstand temporary nutrient stress. And we saw very similar trends when we food and water deprived this stock of flies. Okay. More recently, we've been interested in trying to understand whether these behavioral strategies and increased sleep is something that's stable across the lifespan of these flies. This is an interesting question because the insect life cycle is a little bit different than mammals. The genome basically has to drive the construction of the CNS in almost independently twice, where during development, you can see, so female flies will lay their eggs on a rich food source, which allows their larval offspring to develop and have as many nutrients as they need typically. And then the flies will eclose and come out into a world that's much different than the one that they're raised in. So in order to assess larval behavior, we teamed up with Xu Hao Li, who was a master's student in Matt Kaiser's lab at Penn, and sent them some of our Drosophila melanovensis stocks to assess. They collected larvae and then put individual larvae in wells in a 96 well plate, videotaped their activity, and quantified sleep over a couple of days of larval molting. While our adult experiments found that Drosophila melanovensis show a large increase in sleep compared to melanogaster, we saw the opposite effect with the larvae, where 3 strains of Drosophila melanovensis showed strong sleep reductions compared to CS flies. In contrast, when Matt's group measured feeding in these flies using a dye absorbance assay, they saw a several-fold increase in feeding in their Drosophila melanovensis stocks compared to Drosophila melanogaster. And we think that this is part of a lifelong behavioral adaptation strategy, where larval flies basically trade sleep for feeding, which allows them to build up high metabolic stores in the form of triglycerides that they can then draw down on once they're adults. If we schematize this, we basically see contrasting behavioral strategies at different life stages that are really complementary. Female adult desert flies will lay their eggs in a nice ripe cactus where the larvae can hatch and the flies can suppress sleep and feed as much as they can to pack in nutrients. This allows them to build up fatty acid stores and large nutrient banks. And then once the adult flies eclose, they can most likely live largely off those nutrients that they've already stored up from larval development and hunker down and have high levels of sleep in order to maintain energetic efficiency. So we're really interested in trying to understand the genetic and neural mechanisms that drive these opposing strategies at different life stages, and how a single genome might drive both of these adaptations at once. So hopefully this data convinces you that looking at behavior across Drosophila species might provide a nice platform to understand sleep evolution more broadly. This is just kind of a proof of principle that got us interested in looking more broadly. And so we started to get Drosophila stocks in from different species that originate throughout the world. This is an example of a data set that includes about 60-something different Drosophila species. Just as an illustration, this red shading shows our Drosophila melanogaster stock, and the blue shading shows our Drosophila moavensis stock. To our surprise, we see a lot of fly species that sleep more similarly to moavensis than to melanogaster. But there are a few other shorter sleeping species kind of scattered throughout the phylogenetic tree here. Interestingly, there's a couple of pairs of species, like this one here, where you see really large discrepancies in sleep, and really closely related species. And so we're trying to identify these pairs to dig in more closely and identify both the cellular and molecular mechanisms that might drive these rapid sleep adaptations, and whether if these adaptations arise independently in multiple spots in the phylogenetic tree, if they're driven by the same mechanisms, or if there are independent pathways that occur in each one of these. Importantly, efforts from other labs have assembled high-quality reference genomes for all of these species. And so we're at the point now where we can do bioinformatics studies to try to identify mechanisms that we can dig into in particularly interesting model species, and follow up from there. So we'll wrap up there. Just to summarize, we found that our desert-adapted fly species sleep much more than Drosophila melanogaster, that they homeostatically defend these high sleep amounts, that we've identified brain neurochemistry changes that correlate with the amount of sleep that these different species get. High sleep contributes to resilience during starvation or dehydration in desert-adapted flies, and that their behavioral strategies vary in complementary ways across different life stages. So we're now assessing sleep and behavioral patterns across a larger panel of Drosophila species to try to assess kind of the range of different phenotypes, and then dig in from there to identify genetic and neural mechanisms that drive these behavioral strategies. And we think that this might be a good way to identify adaptive strategies that different species take on to survive and thrive in different stressful environments throughout the world. And so I'll wrap up there and thank everybody who did the work. So Prabhjit Singh took on all of the injury studies that I talked about at the beginning of the talk. Jackie Weiss with May Blundell followed up and did the plasticity studies in the mushroom body and the whole brain. Natalie Moncada is following up on the injury studies now, so stay tuned for those mechanisms. And then for the comparative work, Cesar Nave is a great postdoc in the lab, teamed up with Jessica Yano to characterize Drosophila moavensis, and Cesar is now following up on lots of species to see what the range of behavioral phenotypes are. Okay, and I'd like to thank our collaborators for all of that work, and I'd be happy to take questions if there's time. Thank you.